Hello and good morning, students. This video, um, this recorded video is intended for um, Thursday lesson. Okay, right. We have covered the Venus flytrap. The concept of the Venus flytrap is very important, especially in the closure. Okay, so you don't need to know the opening; it's just the closure. And then, so today's learning objective, we're going to look at the second one, which is explain the role of auxin in elongation. Okay, elongation, growth by stimulating proton pumping to acidify cell wall. Okay. And if we have time, if we have enough time, we will also continue learning objective number 3 and the role of gibberellins, which is in the germination of barley. Okay, so remember this, that auxin, okay, is only in the elongation because there are many roles of auxin. Auxin is can cause can promote the development of roots, can also stimulate the ripening of fruits. There are many roles of auxin. For this particular learning objective or syllabus, it's only for the elongation. Yeah. All right. Let's have a look. Okay, plant growth regulators. So before we talk about all these names, all these names of plant growth regulators, which includes auxin and gibberellins, what is the meaning of plant growth regulators, or also known as plant hormones? So a plant growth regulator is a substance which is produced by the plant and influences growth. Okay, influences growth. So the term that is being used here is influences. So it can promote, it can also prevent. So it actually depends. Okay, influences growth and development. It can stop development. It can also promote or stimulate development. It may bring about its effect near the site of its formation, or it may be transported and act in another part of the plant. Okay, and then definition of growth is a permanent increase in the living organic material of an organism. This involves, what is growth? Production of new cells, increase in organic materials in cells, or increase in the size. So that's the meaning of growth. We will summarize that. Okay, so what's the meaning of growth? New cells, increase in new cells, from 1 cell to 2 cells, 2 cells to 4 cells, so that means growth occurs. Or increase in organic materials, like what it produces, like maybe it produces more enzymes, more sucrose, yeah, in the phloem. So that also means growth. Okay, and then increase in cell size. So cell size, it doesn't mean new more cells. Okay, increase in cell size here, you need to understand the meaning. Increase in cell size means small cell becomes medium size and then large size. Okay, how do plants grow? So plants may grow from our lower 6 syllabus. They grow from certain areas of the plant. For example, what we call meristem. And we talked about this last year. where these are the areas where a lot of mitosis processes are happening. Okay, meristems. So what is meristem? So this is a recap. So meristem are basically specific regions of the plant, like the root tip and shoot tip, where cells are undergoing, cells are dividing, cells are undergoing differentiation, and cells are maturation, undergoing maturation. Yeah. So meristem found at the tips of root and shoots. Okay, and just a recap from your lower 6 here, the zone of cell division, where the shape is still square, it's not elongated, the cellulose is still in its primary stage. Cytoplasm are without vacuoles. Okay, this is in the zone of cell division, and then elongation, and then differentiation. Okay, the same thing with the root tip. Okay, shoot tip and root tip, it has the 3 different zones. The only thing about the root tip is that it has the root cap, which provides protection from the actively growing or dividing cells from becoming damaged as it burrows into the ground. So this is a recap, a recap of so this part here is zone of cell division, zone of elongation, and zone of differentiation. Okay, so you need to know the characteristics in each zone. Here, the cell wall is still in its primary stage, no vacuole. This one is elongated. 2 to 3 vacuoles begin to appear, and then the differentiation phase, large vacuole, and then the cellulose cell wall is already matured. Okay, it's fully stretched. Okay. Right. Let me just inform you or tell you the 5 most common plant growth regulators. So it's a pity that you only learn, so your syllabus is only auxin and gibberellins, and even that, you're not going to learn all of the functions of auxin and all of the functions of gibberellins. Okay, according to your syllabus, you're only required for auxin. You're only required to learn elongation. Okay, how plants elongate, and then for gibberellins, it's only for germination, how seeds germinate. And of course, there are others. You need to know that there are other plant growth regulators you have encountered. We have studied abscisic acid, especially in the closure of stomata. Okay, closure of stomata. Remember abscisic acid, and then ethene gas, and then cytokinin. Okay, so these 3 are not required, except for that abscisic acid in the closure of stomata. Okay, so if you look auxin, okay, we look at auxin here. You see, stimulate stem elongation. Okay, and there are many other roles or function of auxin which is not in the syllabus. Okay, so we are only focusing on stem elongation. How does auxin have an effect? What is the mechanism? How can it explain how auxin can cause stem elongation? And when we look at gibberellins, if we have time, otherwise we'll do the gibberellins on Saturday. We're going to look at the function is in seed development and germination. Okay, so this is the only one which is in your syllabus. Okay, so I'm just going to tell you that there are many functions of these plant growth regulators. Okay, so we're just going to focus on only one as far as the syllabus is concerned, only one for each hormone. Okay, let's look at this one here. I have shown to you the learning objective. Explain the role of auxin in elongation, growth by stimulating proton pumping to acidify cell wall. Now this looks very, very familiar from our yesterday's lesson when we talk about the closure of the venous fly trap. Okay, so the concept is very, very similar, very, very similar. The only difference is the ions that enter. All right. Right. Auxin is also known as indole triacetic acid, or IAA. IAA, or indole triacetic acid. This is another name for auxin. Okay. So what does it say here? It can promote elongation of stem cells. The highest concentration of auxin is produced in the shoot apical. Apical means the tip of shoot, okay, which is the meristem, growing leaves and fruits. So auxin can also be found in growing leaves and fruit. And this is the structure of auxin. Not really required for you to know. And this is just experiment to show that auxin involved in stem elongation, stem growth and elongation. Okay, so if you if you cut the tip here, there is no elongation. But then if you replace back the tip, elongation resumes. Okay, elongation resumes. If you cut the tip, you put it on the agar jelly, so the auxin will diffuse into the agar jelly, and then you put the agar jelly on the this region here, and then it will resume. Elongation will resume. And interestingly, if you put this agar jelly only partial like this, then it will move to the right hand side. That means the auxin moves in a straight line, or diffuse down in a straight line. Okay, so it will curve because this side expand, this area here will expand more, and so it will move to the right hand side. Okay, so the question that we should be asking: how does it do? How does this auxin cause elongation in plant cells? Right, before we talk about the mechanism of this auxin, we need to recap again the structure of cellulose cell wall. We did some recap on this yesterday, where when we talk about the venous flytrap, the venous flytrap. Now we're going to look again. Let's do a recap now. Okay, so first of all, cellulose cell wall. Okay, so if you take any section, if you take any part from here, any section here, then that's called cellulose fiber. Okay, cellulose fiber, just like this one here. And each cellulose fiber, so if we look at one cellulose fiber, so like this one here, this is one cellulose fiber. Okay, see, it has many microfibrils. It has many microfibrils. So each cellulose fiber contains many microfibrils. Okay. So this is the microfibril, another microfibril, another microfibril, another microfibril, yeah. And within each microfibril, you will see that there are lots of this cellulose molecule. Okay. Now this is one cellulose molecule, one long cellulose molecule. In my lower 6 slide, yeah, I also call this one beta glucose chain. Okay, one beta glucose chain. So this is actually one chain. It consists of beta glucose, one chain of beta glucose, or this is a molecule of cellulose. Yeah, just a part. It says here part. Okay, part. So many of this molecule or chain, beta chain, will make up microfibril, and many microfibrils combined together will create many microfibrils combined together will create cellulose fiber. Okay, so this is very important. Okay, as a recap. Right, this is taken from the internet. Just to I was looking for good or best image for this cellulose cell wall. I could only found this one. Okay, right. So this is a microfibril. So here already you have lots of microfibril. Okay, lots of microfibril here, and if we look at one microfibril, we can see there are many cellulose molecule. Okay, or one beta glucose chain. Yeah, one beta glucose chain, another beta glucose chain, another beta glucose chain, or one cellulose molecule, another cellulose molecule. Yeah, and many beta glucose chain will create microfibril, and many microfibril will create cellulose fiber. Okay, cellulose fiber. Okay, so this is a recap. Okay, a recap of the structure of cellulose cell wall. Now, okay, so what happens is that how does elongation, because we say that auxin causes stem elongation or growth, but how does it what is the mechanism? Okay, so initially this is the auxin. The auxin will bind to the receptor. Okay, bind to the receptor, and then it will stimulate the pumping of proton. This is the same like the Venus flytrap. Okay, the same like the Venus flytrap. But instead of the auxin, it is the calcium. Yeah, the calcium. This is in the Venus flytrap. So in the Venus flytrap, it is actually the calcium, the entry of calcium, that starts the pumping. But here the auxin must bind to the receptor. You need to remember the complementary binding. Okay, so proton is being pumped. Okay, proton is being pumped here. So more H plus, concentration of H plus here will increase. Okay, meaning the pH will decrease. Okay, pH will decrease, and so the pH will cause will loosen up the cross link. Yeah, will loosen up the cross link between the cellulose microfibril. Okay, we'll talk about that in a moment more in detail. Okay, but okay, apart from the pumping, let's talk more. Yeah, what does it say? Apart from the pumping, it also stimulates the entry of potassium. It says here, it stimulates the pumping of proton of the cell into the cell wall, where they lower the pH and break bonds. Potassium ion channels are also open to an increase in potassium ion concentration in the cytoplasm. This increases the water potential, so water enters to the aquaporine. Okay, so the binding of the auxin does two important things: the pumping of the protons. Now this will have another effect. Okay, this because the pH will lower. But apart from that, when the auxin binds here, protons are being pumped from the cytoplasm to the cell wall, and then the entry of potassium. So entry of potassium here, high potassium concentration, which means low water potential. Okay, so here high water potential, so now water can pass through even more. Okay, of course water can also pass, water can cross the lipid bilayer. Okay, no problem, but having an aquaporine here will increase the movement of water into the cytoplasm even more. Okay, so you know that when water enters, the cells become enlarged. Okay, and that's when it starts to expand and becomes longer, and that is what we call growth. Okay, and when it expands, when the cell expands, it can expand because the cellulose cell wall is flexible. It's already flexible. The cellulose cell wall is already flexible because of the loosened cross linkages between the cellulose microfibril due to the low pH, due to the high concentration of acid. Okay, now let's talk more. Okay, in the cell walls, the protein expansion. Now this is important because this is mentioned in your syllabus, and then not in the syllabus but in the textbook. Sorry, in your textbook it is mentioned. The expansion is mentioned. So the protein expansions are activated by the decrease in pH. So the pH decreases, that will activate the protein expansion. What does it do here? What does this protein expansion do? Loosen the linkages between cellulose microfibril, thus allowing the cell to expand and elongate. So let me highlight here. It says loosen. Okay, loosen. It doesn't mean break. Yeah, loosen doesn't mean break. Yeah, so I just want to get the concept right, because the breaking of the linkages is done by another enzyme. Okay, another type of enzyme, but the expansion is the one that loosen the linkages between the cellulose microfibril. So loosen the cellulose microfibril means this one here. Okay, this is microfibril. This is one microfibril. This is another one microfibril. This is another one microfibril. So you can see that they are very tight. Yeah, they are very tight here in this one cellulose fiber. Okay, so there we go. So it loosen up. Loosen means able to slide, is able to slide past one another, is able to move past one another, and so which allows the cell to elongate and expand as it absorbs water, and because of that, elongation can occur. Okay. Right, now this is from the same images that I provided yesterday with the venous flytrap, except that we are not dealing with with the venous flytrap. It's just calcium. Yeah, entry of calcium causes the pumping of the proton. So if we look back, just want to have a clear, you must be able to differentiate, yeah, between the two. Okay, as you can see here, because of the calcium, the entry of the calcium causes action potential, and it starts to pump the proton. Pumps the proton, acid drop, cell wall crosslink broken. So this is the same, yeah? The concept of cell wall crosslink broken is the same as in the auxin. We're talking about the hemicellulose, yeah? So I did show you this one yesterday. Okay? So this is the one that is being, initially it's loosened up and then it will be broken. That is in the case of auxin, yeah? This is the one that will be broken. The hemicellulose, this one here, hemicellulose, okay, the green color here. The same thing with here, but except that I provided you this with this image, but I put the auxin here. I told you this is a different syllabus, okay, but now it is related. Okay, so the breaking of the crosslink is the same. It's actually breaking the hemicellulose. Okay, so it's easy now to, the difference is only this is the binding of auxin, whereas that one is the influx of calcium causes action potential and so forth. Okay, so you need to differentiate that too. All right. Now, so the crosslink is actually by the hemicellulose. So I modified this slide. This is the crosslink polysaccharide. So hemicellulose is a type of polysaccharide. Okay, it's a polymer. It's a type of polysaccharide. So it actually making crosslink. See here, making the crosslink. This is one microfibril. This is another microfibril, and there is a crosslink. Crosslink means there is a structure or a component that links these two microfibril. Okay, so the acid, the entry of this acid here, the pumping of this acid here will loosen this crosslink. It will loosen this crosslink. Loosen means, what is the meaning of loosen? Loosen means, in the as far as the textbook is concerned, this gets separated. This linkage separated from the microfibril. So when it is separated like this, so this area is exposed. Yeah, this area is exposed. It is exposed to be broken down by this enzyme. So this is the enzyme. It is not known what the enzyme is. It is not known what the enzyme is, but when this is exposed, then the enzyme can cut the crosslink. So when it is being cut, then the cellulose cell wall now becomes flexible. So the concept is the same. Yeah, is the same with the venous flytrap. Okay, if we check again, the concept is the same with the venous flytrap. As you can see here, pumping of the H plus from cytoplasm to the cell wall, and then the cell wall loosen, crosslink will eventually be broken. Okay, and the cell wall becomes flexible, and now water can enter, and then it will shut, or it will close, the trap will be closed or will be shut. So it's like that. It is the same, yeah, almost the same, except that in this learning objective we mentioned the term expansin. In the venous flytrap, it doesn't mention the term expansin. so there has to be some limits in the explanation. Okay, so again, let me repeat about this. What does the acid do? It loosens the linkage, the crosslink between cellulose microfibril. Okay, this is one microfibril, another microfibril, loosen, and then this is exposed. This part is exposed, and when it is exposed, you can see this enzyme will cut it, cleave. Yeah. Here, cleave Cleave also means cut. Okay, break. All right, this is from a university textbook. Yeah, so I put a university textbook here just to make things more, to get the concept right. It's very important to get the concept right. Okay, first of all, remember the pumping requires proton energy. Sorry, the pumping requires the hydrolysis of ATP. Now, this is highlighted here. I purposely highlight this. You see, wage. Let's read this one. This is from a university textbook, so it's very, very. It has high credibility. Yeah. So wage shape expansin, red. So this is the expansin. What is expansin? Expansin is a protein. It's not an enzyme. It's just a protein that loosen up the linkages. Okay. Activated by low pH, separate cellulose microfibril from the green. So this separate from the green. Now you can see it's attached here, so it will be separated. It will be separated so that this area here is exposed. Yeah. The exposed, see. So now they use the word exposed, cross-linked. So I presume, okay, this is just my assumption, is that previously the cross-link is available. There is presence of cross-link, but maybe it's very, very, the distance is very narrow. Yeah, there's no gap, or the gap is very, very little gap or limited. But because of the expansin is activated by the low pH, the gap is now widened up, and therefore it exposed the cross-linked. So when the cross-link is exposed, now the enzyme here can cut. Okay, you see here the cellulose loosening enzyme purple cleave or cut the cross-linked, allowing cellulose microfibrils to slide. The extensibility of the cell is increased. Trigger causes the cell to expand. Okay, so that's it. Yeah, that's as far as this syllabus is concerned. So actually, when you look at it, we actually comprehensively answer. We actually comprehensively tackle this learning objective, yeah, to the T, meaning to the very, very detailed, from, very, very detailed. Okay, so now just a few definition. What is hemicellulose? The hemicellulose is the cross-linked. Yeah, remember, this is the hemicellulose, the green color here. This is the cross-linked. Okay, what is it? What is this hemicellulose? This is taken from biology Oxford Dictionary. Okay, Oxford Dictionary. Okay, so it has high credibility. You can trust this information. It is a polysaccharide. So it's a polysaccharide found in cell, plant wall of plants. The branch chain of this molecule bind to cellulose microfibril together with pectin, which is not really important in this case, forming a network of cross-linked. Yeah, so the cross-link is actually the hemicellulose. This is the name of the cross-link, but the syllabus doesn't mention to you. So I feel like that it's very nice. It's good to know the name of the cross-linked. Okay, so let's read this one here. Molecules of auxin bind to a receptor protein on the cell surface membrane. The binding of auxin stimulates ATPase protein pump to move hydrogen ions. Okay, protein known as expansin. So this is not an enzyme. Yeah? This is just a loosening protein. Is activated by decrease in pH. Loosen. So it's very important that this is not. It doesn't break. The concept you get the concept right. It loosens the linkages between cellulose microfibril. It is not known exactly how they do this, but it is thought that expansin disrupt the non-covalent interaction between the cellulose microfibrils and the hemicelluloses, which is the cross-link. This disruption occurs briefly so that microfibrils can move past each other, allowing the cell to expand without losing much of the overall strength of the wall. Okay, so very, very important. These two slides, these three, I mean, this picture here, this one, this one, this one, and this one, you need to master this just for one learning objective. Okay, now what about expansin? This is also taken from Oxford Dictionary. Okay, it's not something that I got it from online. It is from I have my own dictionary, a biology dictionary, Oxford, and this is what it says. It is a leucine, enabling expansion of the cell during growth and other development. They are thought to act by weakening the non-covalent bonds between cellulose microfibrils. So there is a bond which is non-covalent, okay? That is not really important what bonds, allowing the latter to slide relative to each other without disrupting the cell wall. Okay, this is the sequence. This is from Bradfield textbook, but there's a lot of missing, not missing, it's just that it lacks one or two important points, but it can still be used, yeah? It is enough, I guess, to score 4 or 5 marks. So auxin bind to the receptor, activates protein pumping, okay, from cytoplasm to cell wall, lowers the pH, okay. Now this will activate, so this is the part that it doesn't mention. This should say will activate expansin, okay, expansin. And then loosening the cross linkages between cellulose microfibril. Okay, so loosening. Then once the cross-link is exposed, then the enzyme, which we don't know what the enzyme is, then the enzyme can break. Yeah, it says here enzymes to act which break bonds between adjacent cellulose microfibrils, which actually breaking the hemicelluloses. The microfibrils can slide past each other and keep this wall flexible. And then the cell absorb water by osmosis, and then it can expand and then start to elongate. Okay. All right. So this is again. This is the last. This is the last slide. Yeah. This is the last slide. Again, let's have a look. Auxin diffuse away from the tip, binds the receptor, activates hydrogen pump, lower the pH. So remember, the low pH activates the expansin, okay, loosening the linkage, the cross-link between cellulose microfibril, and then the enzyme will break the cross-linked, and then when it absorbs water, when this cell absorbs water, these are all cells, yeah. These cells absorb water. It only occurs in the meristem, okay, shoot tip or root tip. Then it can expand, and that's how growth occurs. Expand, yeah. The expansion, the elongation. How can it elongate? Two things, because now the cell wall is flexible, and it can also elongate because it absorbs water. Okay. All right, so that's it. Yeah, I think that's it for Thursday lesson. I think the Gabrielians will continue on Saturday. Okay, commercial uses of auxin. This is just not in your syllabus, but there are many other functions of auxin. Yeah, such as rooting powder, meaning to promote development of roots, weed killers. Yeah, selective weed killers. It doesn't kill grass, but it kills the dicotyledonous plant, yeah, like this one here. So auxin actually kills dicot plants here. So this is important, like for example, if you are football field maintenance, for example, because you want only grass to grow, you don't want dicots to grow, so you can apply auxin to kill the dicot plants. And in fruit setting. Sorry, fruit setting means to allow the fruits to stay longer, to last longer. Sorry, last longer. Just spray auxin, it will last longer, and as well as in micro propagation. Micro propagation is not really now, it's not in the syllabus anymore. So on Saturday we'll be talking about gibberellins. So basically, it's very, very important. You just need to have a clear distinction or differentiation between the closure of the venous flytrap with the stem elongation triggered by auxin. Yeah, but there are lots of similarity between the two. Yeah, a lot of similarity, only minor differences. For example, in the venous flytrap, it is the entry of calcium, the influx of calcium, and then action potential occurs, and then it starts to pump the proton into the cell wall. And then the same thing, the cell wall cross-link loosen, and then will eventually be broken. Okay, the same thing. And when it comes to auxin, then you need to talk more. You need to talk about the expansin, what does it do, and then as well as the enzyme. Okay, the expansin, which is loosening the crosslink, okay, of this crosslink between the cellulose microfibril, and then eventually when it is exposed, the gap is now widened, is exposed. Now the enzyme can break the cross linkages. Okay, so that's all for today. I'll be uploading again for Saturday's lesson. Thank you.